This is a production of Cornell University.
Okay, good afternoon. Thanks, Leo, for your introduction. Before I get started, I would like to say a few words for this seminar. That is, I will be giving it as an extension seminar. So, my extension program targets at two groups of audience, the apple industry and the general public, especially the master gardener volunteers. So today, I will be treating you guys as part of these two groups for next hour. So with that said, here's my outline of my talk today. I will be basically addressing two questions. The first is, what is genomics? I'll be beginning with a few key concepts in genomics, and then talk about major accomplishments in genomics. The second question is, why genomics is a vital solution for challenges in fruit and vegetable production. So this is the focus of my talk today. I'll be mentioning briefly the challenges we're facing, and then describe the solutions or genomic solutions for these challenges. I'll be using cabbage black rot resistance and apple fruit quality as example. So before I get started, okay, the first question is about what is genomics. So now to answer the question, let's look at this picture first. Okay, this one is really a touching story. The slide itself is adopted from the fly of a French documentary made a couple of years ago. The gorilla named Dept lived with the family, Pierre and Annie, for 4 years. The gorilla really become a member of the family. But in the end, the gorilla has to leave back to where she really belongs. Because the fully grown gorilla is really different from humans. Now, from the point of genetics, the difference is caused by the genome. Okay, so in other words, we can say that it is the genome that makes this family must be broken off. So, but please do not think a genome is a bad thing. Actually, it is also the genome that make us who we are, why we have such a diverse world. For the same reason, the apple fruits can be always distinguishable from other fruits, such as pear and peach. So you may think it's easy to understand the difference between the species are caused by the genome. But what about the difference within a same genome? Well, that would be also caused by the genome as well? The answer is yes. So for example, these are the 4 types of vegetables. Okay? But they belong to the same species called Brassica rapa. If you look at how they differ from each other. But if you look at their genome sequence, probably more than 99%, they are the same. But the 1% difference is enough for what you see here. So this said that the genome not only determines the difference between the species, but also determines the difference within the same species. A little variation in the genome that can make a huge difference in their appearance. Okay, talk about the genome. What is genome exactly? Okay, in the scientific term, genome is the total collection of DNA or genes in a reproductive cell, such as sperm, egg, pollen, that contains all of the genetic information needed to build an organism. So a genome is really like a blueprint for building a magnificent building, such an empire building we're all familiar with. Now, the DNA of genome contains a lot of millions of bases. So if you print the genome, there will be a hugely thick book. So in the apple genome, there are about 750 million bases. So the apple genome book would have 750 million letters. Assuming we can print 3,000 letters per page, that would count for 250,000 pages. Now, in the genome of an apple cabbage, the genome book will be a little bit thinner, with 284 million letters, but it still takes about 95,000 pages to print it all out. So in the living cells, the genome DNA is naturally divided into a limited number of units and organized into a structure called chromosomes, as shown in this picture. So the apple genome has 17 chromosomes. If we're using the concept of book, so the apple genome book would have 17 chapters. Now, similarly, for the Brassica napa, which is the rubber cabbage, so they have 10 chromosomes. So the Brassica rubber cabbage genome book would have 10 chapters. Now, gene is another important concept in genomics. I think you have heard about it. So genes are the basic functional unit of genome. Genes carry important genetic information. So genes are located at various locations along this chromosome. So a single genome can be a home for genes of thousands. The text box here actually showed a DNA sequence for a gene. Actually, this is the gene responsible for controlling apple fruit acidity we identified recently. So the first three letters, ATG, serves as like a start button for the gene get translated, and the last three letters serves for the stop button for translation termination. The mother letter somehow changed the basis in the middle of the red G, the G1455 we called it, to A1455 in some of the apple varieties. That makes those apples tasteless because the acidity is too low. So now, with the introduction to genome, now let's talk about what is genomics. Okay, genome in the short word is a study of the genome. It's mainly investigating the structure and the function of the genome or whole genome. Okay, depending on the area of focus, there are structural genomics, functional genomics, comparative genomics, and synthetic genomics, and many others. So genomics is a new and dominating discipline in genetics. It's part of genetics. Much has been accomplished in fruit and vegetable genomics. This slide shows about 17 fruits and vegetables which genome has been sequenced, including apple and grape and oranges and tomato. So I want to mention that sequencing of the tomato genome is primarily led by Jim Giovanni here at BTI. The development of talents-based biotechnology is another major accomplishment in genomics. So talents stand for, the word a little bit awkward here, stand for transcription activator-like effector nucleases. Talents-based biotechnology is a major breakthrough in biotechnology that come out from the genomic studies. The major property of talent is to make a cut in the genome at a specific location. So talent is extremely useful and a versatile tool for genome editing because of that property. The major advantage of talent-based biotechnology is that talents improved vegetables and fruits are very comparable with those that are developed by conventional breeding because there's no foreign DNA involved. I'll come back to all this a little bit later too. So the bottom drawing shows a talent that cut a targeted site in the genome. Okay, so much for the introduction of the genome and genomics. So now let's address the second question: why is genomics a vital solution for challenges in fruit and vegetable production? It's always challenging in production of fruits and vegetables nowadays. So in addition to the rising labor cost and the material cost, profitability and pest control are the major challenges. To address the profitability challenges, optimizing cropping system and practice for high yield and quality are commonly used. To control the damaging diseases and pests and insects, the chemical sprays are used most frequently. Are there any other means that are more cost effective? What about developing superior varieties of fruits or vegetables that are naturally of high yield and quality and resistance to multiple diseases and insects? From any standard, this is likely the best approach. This is why genomics can provide a vital tool, vital solution for the challenges we are facing in fruit and vegetable production. There are two ways genomics can provide solutions for developing such superior varieties. The first will be using biotechnology. So given the controversy of existing biotechnology, the talents-based biotechnology really holds the promise. I can see lot long away, we will go breeding by design. The second would be help conventional breeding by marker-assisted breeding or genomic selection. So I'll be now showing with examples. The first example is improvement of black rot resistance in Brassica oleracea. Black rot is probably the most damaging disease in crucifers worldwide. And the causal agent is bacterium Rhamnosus compestris. Black rot is especially damaging to Brassica oleracea vegetables, such as cabbage. So the photo on the lower left side shows the black rot damage to cabbages in the field, and the photo on the lower right side shows the black rot damage inside a cabbage. I got this photo from Griff. Thanks for his help. In addition to cabbage, there are several common vegetables that are also Brassica oleracea, including broccoli and cauliflower and Brussels sprouts and several others shown here. So all of these Brassica oleracea vegetables have problem of black rot disease problem. So what makes black rot a particular problem for Brassica oleracea? There are at least 3 factors that are contributing to this situation. The first is the source of black rot resistance in Brassica oleracea is very rare. There are only about 2 accessions reported of resistance, as shown here. The second, the resistance is very race specific and race dependent. The third factor is the resistance can only be partially bred into new cultivars. So because of these limitations, breeding for high resistance, high black rot resistance has met with little success. To help with this long struggle for high black rot resistance, 2 talents-based approach are envisioned. The first is to identify the resistant genes from Brassica relatives and then transfer them into Brassica oleracea. So the second is to directly modify the susceptibility genes within Brassica oleracea to convert that into resistant genes. So for the first approach, we need to know what the resistance are there in the Brassica relatives. Why the resistance in Brassica relatives cannot be bred into Brassica oleracea. Now here shows strong resistance exists in several Brassica species, including Brassica rapa, Brassica nigra, Brassica jonesii, and Brassica carinata. But the resistance in these relatives cannot be easily bred into Brassica oleracea. To explain that, let's look at the relationships between the Brassica species. There are 6 major Brassica species, including 3 diploids and 3 tetraploids. The 3 diploids are Brassica rapa of A genome of 10 chromosomes, and Brassica nigra of B genome of 8 chromosomes, and Brassica oleracea of C genome of 9 chromosomes. So the 3 tetraploid species are thought to be derived from the natural hybridization between the diploid species, followed by chromosome doubling. The genome of lava cabbage, which is we call Brassica RAPA has been completely sequenced, and the Brassica oleracea certainly is the topic of this example. Now, if you make a cross between Brassica oleracea with any of these species with resistance, the hybrid will be not viable, because the imbalance of chromosome numbers. So this explains why the resistance in other Brassica species cannot be moved into Brassica oleracea by conventional breeding. Therefore, talents-based biotechnology is an ideal choice for moving the Brassica native resistant gene into Brassica oleracea. The second approach is more exciting because we don't need to involve any other Brassica species. We simply need to identify the susceptibility genes. This approach has been recently demonstrated successfully in rice. I'll show you in next slides. Now, the first row shows a part of a gene in a rice cultivar that is susceptible to bacterial blight disease, which is a most destructive disease in rice, one of them. Now, the rest of the rows basically is the mutations that are induced by talents. Okay, the dash stand for the DNA that were missing, that were deleted by the talents, and the letter in the red are the DNA bases added by the talents. Now, here's a comparison of the difference in the niche length between the non-modified control and the talents modified rice line. So it's clear that the susceptibility gene has been converted into resistant genes. As I said, the talents modified rice line contains no foreign DNA. So these rice lines are really comparable with conventionally bred any rice varieties. So with talents-based approach, high black rot resistance in Brassica oleracea will be available soon. So this is so much for the first example. Now I'm going to talk about the second example, which is the improvement of fruit quality based on the fruit acidity. I will show you how marker-assisted breeding has been done or can be done to improve fruit quality. There's two major reasons to begin with apple fruit acidity. The first reason is that fruit acidity largely determines fruit taste and flavor. So fruit acidity is one of the major components that affect fruit quality. So other components include crispness, firmness, juiciness, size, soluble content, storability, and texture. And the second reason, probably the major reason, the apple breeders rate fruit acidity the #1 priority trait for breeding and for genetic studies. I'll come back to this point, why apple breeders rated fruit acidity the #1 priority trait. Apple varies widely in the fruit acidity levels. Based on our own measurement, fruit pH varies from 2.8 to 4.5, and treatable acidity, the TA, varies from 0.07% to 0.187%. The acceptable range for a dessert apple variety is pH 3.1 to to 3.8, or Ta 0.3 to 1%. Apple fruits with fruit acidity levels beyond this range are either too high or too low in acidity. In apple genetics, it's known that acidity levels is primarily controlled by the gene we call MA, as I mentioned earlier. There are two alleles or versions of this gene, one for high acid, one for low acid. The high acid allele is dominant over the low acid allele. So there are three possible genotypes in apple. Homozygous high acid genotype, and heterozygous high acid genotype, and homozygous low acid genotype. Because apple received one allele from the seed parent, another allele from the pollen. In a survey, 79% of the named apple varieties are heterozygous high acid genotype, and 14% homozygous high acid genotype, and 7% homozygous low acid genotype. So these two high acid genotypes supposed to produce fruits of high acid, and the low acid genotype produce low acid fruit. When the apple has a low acid genotype, the fruit tastes very flat, no flavor. So in breeding programs, those seedlings will be useless, no commercial value, need to be thrown away as quickly as possible. Since the heterozygous high acid genotype is dominant in the named apple varieties, so most breeding population is derived from two genotypes that are both heterozygous high acid, as shown here. So the progenies of this kind of crosses surrogates like 1/4 of seedlings of low acid genotype. So although this breeder knows that, okay, growing these genotypes is useless, it's wasting time, money, but they cannot throw them away with confidence. That's why they rate fruit acidity as the #1 priority trait. If someone can help them doing this one, that'd be great. So that's why we wanted to identify the fruit acidity gene. So through extensive effort, we were able to reveal the identity of gene MA. As I mentioned earlier, the MA gene has 1,707 DNA bases, and encoding a malic acid transporter protein of 568 amino acids. The arrow stands for the radial direction of the gene. Okay, the upper two rows shows the DNA bases and their corresponding positions, with the omission of the majority. Okay, the lower two rows shows the corresponding amino acid residues and the corresponding position. We find that a single base mutation from G1455 to A1455 in the gene MA is responsible for low acid fruit, as I mentioned earlier. The reason is that the mutation caused a premature stop button in the MA gene, as shown here. This is a mutation, then it caused this stop button here, so then they cut off the rest of the amino acids, which is 84 of them. So presumably, these cause the MA gene malfunction. So with the finding of this gene, we want to use it in apple breeding. Now, we developed a marker called CAPS1455 to specifically targeting the mutation site at the location. Now, this gel shows a example of analysis of the marker CAPS 1455 in a diverse set of apple varieties and selections. Each of the lanes stands for apple variety or a selection. The top band stands for high acid allele. The lower band stands for low acid allele. The single top bands in these lanes are for homozygous high acid in red, and the single lower bands in the lanes stand for homozygous low acid genotype. And those with two bands stand for heterozygous high acid genotype. So those in red and in yellow are expected to produce fruit of high acid, and those in white are expected to produce fruit of low acid. Here is the pH data for those varieties and selections. This is exactly what we expected. For example, in the three white lanes, which is a homozygous low acid genotype, the pH measured from 4.3 to 4.5. Very high pH, suggesting low acid. So this indicating that marker CAPS 1455 is a versatile marker for marker-assisted breeding. How to apply this marker in apple breeding? So the first will be marker-assisted selection of siblings. So in collaboration with apple breeder, Dr. Susan Brown, we test this marker in one of her large apple breeding population. Again, each lane stands for a sibling. So this gel picture is just showing part of the results. Now, the lane with a single band that indicated with a red arrow are for homozygous high acid genotype. And those with the two bands indicated with a yellow arrow are heterozygous high acid genotype. And those with a single lower band are homozygous low acid genotype. So just based on, we have 96 siblings shown here. So we have about 23% of low acid genotype. It's very close to 25% as we expected. So those siblings theoretically can be thrown away. I have suggested Susan do so, but she hasn't. So the second approach, the second application is marker-assisted planning for new crosses. Now, the idea is to maximize the heterozygous high acid genotype in the progeny while minimize the portion of homozygous low acid genotype. So the base cross will be made between a homozygous low acid genotype and a homozygous high acid genotype, like shown here with dark green, because progeny of these crosses are 100% heterozygous high acid genotype. So the cross is between two heterozygous high acid genotype or between a heterozygous high acid genotype with a homozygous high acid genotype is also acceptable. And the cross is made between a homozygous low acid genotype with a heterozygous high acid genotype. That can be done, but half of that, 50% of the siblings will be segregated for low acid genotype. So crosses to avoid are those between two homozygous low acid genotype and two homozygous high acid genotype. Okay, in summary, so genome is the total collection of DNA or genes in reproductive cells. The genomics is a study of the structural function of the whole genome. TALENS represents one of the major breakthroughs in genome editing and in precise and clean genetic engineering. And genomics provides vital solutions for challenges that cannot be effectively addressed through conventional breeding. Genomics helps increase breeding efficiency, especially in tree fruits of a long juvenile phase. I would thank Dr. Susan Brown, Lilian Chen, and Gennaro Fado for their help in the fruit acidity project. Also would like to thank Debbie and Laurie for help in my extension program. Yanbei is my grad student, and Laura is a technician in the lab. So I would like to stop here. Thank you for attention and ready for any questions. This is a very nice question. That's the question actually I ask myself all the time. Actually, I didn't show here. When we try to identify the gene MA, we genotyped many apple seedlings in the population. So if you look at their distribution, so clearly there's two peaks. That's the breaking point around 3.8 in pH, and the TA is around 0.3. So that's why we put one category as low acid, another category with a high acid. Now, you're absolutely right. So there's a continuous variation within either of the categories. So I think this is the next phase of our goal to find out what exactly causing that. But the MA gene, the major function of the MA gene is to make that curve, okay, for the high acid group and for the low acid group. Now, to fine tuning it, certainly there's other factors involved. That's the things we are pursuing very vigorously. When I have that, I'll let you know. Yes. Well, I don't mean to discount the rules of cropping system and the orchard or the field management. No, this is not my point. I realize these are very important efforts need to be done, need to be in place to manage the field or the orchard. Now, the point that I try to make is if we have a resistant variety, that will be really helpful. And that's not all, certainly not all. I agree with that. Thank you. I think this will be helpful. I don't know what really the process will be in the regulation, but I would say that based on the fact, okay, there's no foreign DNA being involved in those TALENS modified lines, they should not be regulated at all. This is my view. But people certainly can argue the process that you used is similar to those that we use for conventional biotechnology. But the fact is we don't have anything left. So we must respect this fact. I think it should be easier. Okay, I don't know actually. Maybe I should say first. Then my guess it should be easier. Then your second question, can you remind me that? I forget. Oh, public acceptance. I think that's probably very similar to the first situation. We just need to get the fact out. This is really different from the first generation technology. The technology is different. because there's nothing that really knows nothing being involved in the whole genome. So why you want to reject this if that then turned out to be resistant or turned out to be high yield or high quality? But again, so we can ever know what the public really react to that technology. Well, that. To be honest with you, that's something unexpected. I don't expect that kind of message will be conveyed through that slide. I just want to make a point that it is the genome that make who we are, what the gorilla is, why gorilla is a gorilla. I see your point. Okay, I will pay attention to that. Try to avoid that. Next talk. Okay. Thank you for the comment, Gary. I have a slide prepared for this seminar. Then I'll take it out. It's something called central dogma. The DNA makes RNA makes protein. No, eventually I elect to take that one out. So I may consider to put it back in. Thank you. Well, the collection is this. So when you start in course, try to develop a new apple varieties, you basically don't want those low seedlings in your breeding population. So you want to get rid of those as early as possible to save some space in the orchard. So certainly they'll be reducing some of the cost, hopefully increase the breeding efficiency because you can plant more useful seedlings in a limited area with limited resources. So that's the point. I do not think the 80% apple varieties now have a high acid. Now these are already developed. So what I'm talking about is for getting the better varieties to sooner and quicker and better. Thank you, Larry. My experience with master gardeners are very positive. So looks like they are very they are very receptive to this kind of new information. Certainly, my experience is also limited. Okay, just based on what the feedback that I get. Now, the experience with Apple industry is a little bit different. So let me just show you. Let me just tell you, one of the feedback that I get for the two, basically for the two seminars that two lectures that are given in the winter fruit schools for apple growers. So they scored like 1 is the worst, 4 is the best. Okay, so basically 4 is excellent, 3 is good, and 2 is fair, and 1 is poor. So for the topic itself, I got score 2.5. That's the audience. That's about 400 audience. The feedback is probably more than 100. So that's well, you know where that's where you. Okay.
So the master gardener in the gardening public do not seem to be against the idea of transgenic apples or transgenic cabbages or anything like that.
I do not sense that. Because when I give them a talk, I didn't say the transgenics will be used immediately. Certainly, we need to go through the process. I just try to explain the fact the new technology are there. Things will getting cleaner, so hopefully it will be more acceptable to them. No, it's we look at their acid about 3.7, still within high acid range. In pH, the TA is like TA like a 4, so it's still within the bigger category of high acid. We also have the genotype data using the markers we developed. Fuji is clearly the heterozygous and the Lucas. Yeah.
As an apple grower, I think we're living in one of the parts of the world where people have the greatest preference for tart varieties, for high acid or medium acid apples. And my sense is in most other parts of the world, except maybe Western Europe, people really prefer the lower acid end of the range. So I guess my question is, what's the cutoff? You know, what we know that if an apple, when the firmness gets below 1,000 pounds or something, it'll taste mealy. But what's the cutoff for acidity?
For example, Golden Delicious probably is the most popular apple variety in Europe. Now, if you look at for pH and TA at the harvest, that's way above the border that I said. I said the pH 3.8 and the TA 3.0 or 0.3% is way above the range. So way into the high acid range. What I'm talking about high acid is not really you can't even test. It's basically that's a relative term to those double, basically homologous low acid genotype. Okay.
Let's not thank our speaker.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.